Hello, my name is Gilbert. When this video was made, I am a postdoctoral researcher in the Department of Bioscience Technology at Chung-Der Christian University, Taiwan. In this video, I would like to briefly explain one of my research with title Acute and Chronic Effects of Fin Amputation on Behavior Performance of Adult Zebrafish in 3D Locomotion Test assessed with fractal dimension and entropy analyses and their relationship to fin regeneration. Without further ado, let us get started. Fish need to coordinate movements across their body in capturing prey, avoiding predators, and navigating their environment. This coordination involves fluid movements of the jaws, eyes, tail, and fins. There are a wide diversity of locomotor behaviors of fish, such as rapid sea-start escape response, steady undulatory locomotion, and linear accelerations. However, generally, it is subdivided into swimming with paired or median fins or with the body axis and caudal fin. In zebrafish, paired fins include the pectoral and pelvic fins, while the dorsal, caudal, and anal fins, internal support elements, and external supports belong to median fins. The pectoral fins of juveniles and adults of many fish species act as primary propulsors during rhythmic swimming and in arrhythmic movements such as braking and maneuvering. Tucked pectoral fins along the sides of the body and axial movement alone can generate propulsive thrust. Pelvic fins, the other paired fins, power corrective forces in actinopterygian steady-state swimming. Next, as median fins, dorsal fins increase the lateral surface of the fish's body during swimming. Meanwhile, intermittent caudal and anal fins assist the pectoral fin locomotion to achieve higher relative speeds and provide stability of fish, respectively. In addition, the caudal fin is often observed to sustain damage because of predation, disease, or social rank. Therefore, damage to fish's fins might affect their locomotor performance and compromise their survivability. Despite the importance of fins in fish, unfortunately, very few studies have evaluated in detail the effect of fins amputation on fish locomotor activity, especially zebrafish. This might be because, in most of these studies, the interpretation of the results is very challenging, since the power of the fish to compensate for such deficiencies after amputations by suitable action of the remaining fins on the general equilibrium is so great. Moreover, most of the studies mainly focused only on a single or several fins. Nevertheless, several studies have built a foundation to highlight the significance of fins in fish locomotion. For example, using sockeye salmon as an animal model, a previous experiment examined drag and thrust energetic relations for a swimming fish after caudal fin amputation. From the results, a significant reduction in the critical speed of fish was observed when both apaxial and hypaxial caudal fin lobes were removed. Furthermore, a prior study compared the swimming performances of 8 groups of rainbow trout with various fin rays removed. Later, significant differences in the performances between every group were observed, confirming the hydromechanical theory that large fins are required for acceleration. In addition, research on American dogfish demonstrated the instability of the equilibrium in the horizontal and vertical planes without fins by using the wind tunnel. Meanwhile, in the vertical plane, the instability is significantly increased by the presence of the pectorals, indicating the significance of fins for the fish to achieve the normal equilibrium of swimming. However, to the best of our knowledge, there has been no study comprehensively comparing the differences between each amputated fin in altering the locomotion of adult zebrafish. In recent years, video-based automatic methods have been broadly applied to monitor the behaviors of aquatic animals. These methods actively replace manual observation due to their high overall validity, objectivity, and consistency of collected data, and the number of assessed behavioral endpoints. Furthermore, the characteristics of these methods make them suitable for extracting neurobehavioral phenotypes of zebrafish, since, unlike rodents and humans, the locomotion of zebrafish and many other aquatic animals occurs in 3D coordinates, generating complex behaviors and multidimensional datasets. Since their biological activities are complex and inconsistent, conventional methods such as Euclidean geometry are insufficient to describe them. Therefore, together with entropy analysis, complex geometry such as fractal geometry is required. Fractal dimensions and entropies are usually utilized to measure complex systems in the real world. Fractal dimensions characterize fractal patterns by featuring the complexity as a ratio of the change in detail to the change proportionally. Lately, this measurement has been used to describe behavioral patterns of various animal models, including fruit flies and chironomids. In zebrafish, FD reduces the 3D complex swimming trajectories of zebrafish to one value and was used in a previous study to quantify their swimming behavior provoked by the presence of hypochlorite in the water by using FD analysis. It has also been applied to various painful treatments in zebrafish to produce an arbitrary pain intensity scale. However, even though FD is used for behavioral monitoring, it mainly presents the overall trend of behavioral changes that are limited in presenting global and local information of movement changes after exposure to stimuli. Therefore, entropy, which is also a nonlinear measurement to describe the uncertainty and degree of disorder, is used to help the researchers in observing the behavior changes from different perspectives since it is useful for detecting changes in behavioral states overall and in specific changes including the transient periods under stressful conditions. In addition, it also has been applied in complex biological systems. Permutation entropy has been proven helpful in a wide range of applications where the measurement of complex time series were needed. For example, in a prior publication, it was applied to analyze the behavioral changes of the fruit fly in response to stressors. We believe that combining these approaches can give different perspectives from the commonly used behavior endpoints in observing zebrafish behavior alterations after treatments. Addressing the significance of the fins of fish, we investigate the effects of several fins amputation on fish locomotion by combining a 3D locomotion video-based automatic method, fractal dimensional, and entropy calculation from the continuous data to reflect changes in movement behaviors. Using this combined approach, this research is the first to study detailed three-dimensional movements of fin-amputated fish and evaluate hypotheses for how these fins might function. Later, based on those data, we found that the absence of one type of fins caused more severe behavior alterations in the fishes than other fins. Afterward, we conducted a more profound investigation regarding their recovery process after amputating this fin in terms of their behaviors in the 3D locomotion test. In addition, the potential cause of the behavior alterations was also studied by using lidocaine, a local anesthetic with analgesic properties. A three-dimensional locomotor activity test is a sensitive tool to assess the alterations in zebrafish swimming behavior after treatments. Here, the swimming behaviors of the fin-amputated fishes in a group were observed 2 days post-amputation. From the results, amputations of fins affected the swimming behaviors of zebrafish. In terms of locomotion, the absence of all of the fins reduced zebrafish locomotion. Meanwhile, regarding their movement orientation, even though none of the amputated groups showed a statistical difference in the average angular velocity, except for the caudal fin amputated group, the amputations affected the turning movement of the zebrafish. Next, similar to the locomotion results, amputations of fins led the fish to exhibit abnormal exploratory behaviors in different manners. To further explore the effects of fin amputations on zebrafish swimming pattern, the zebrafish swimming behaviors in the 3D locomotor activity test was also observed individually since zebrafish possess different behaviors in the presence of conspecifics. Interestingly, amputations in all fins did not alter their swimming pattern, especially in locomotor activity and exploratory behaviors. In terms of the results from FD and entropy analyses, statistical decrement was shown in the FD between control and several amputated groups, including caudal, pelvic, and pectoral fins in grouped zebrafish test, while this phenomenon was not displayed in the individual test. Meanwhile, only the caudal fin amputated group showed a statistically higher level of entropy than the control group, demonstrating the effect of this fin amputation on the zebrafish response. PCA and heat map analyses were also carried out to reduce the data dimension and complexity, thus allowing us to explore the behavioral phenomics between every group. From both analyses, all experimental groups were divided into 2 major clusters. In the first cluster, anal and dorsal fin amputated groups were found to belong in this cluster, including the untreated group, indicating that these 2 amputated groups exhibited more similar behaviors to the control group than other treated groups, including caudal fin amputated group. In conclusion, the caudal fin amputation had the most significant effects on the zebrafish behavior performance in the 3D locomotion test, even at 2 dpa. Therefore, it is intriguing to conduct a more profound study regarding the relationship between their behavior recovery after their caudal fin is amputated and their caudal fin regeneration. Since the absence of a caudal fin was found to affect the zebrafish behaviors more severely than other fins even after 48 hours of recovery, it is intriguing to understand how early their behaviors are recovered after the amputation. Therefore, a periodic observation was conducted on the amputated zebrafish by the group 3D locomotor activity test. From the results, in terms of locomotion, the behavior recovery was approximately started at 1 dpa, with almost complete recovery on day 5 and full recovery on day 10. Interestingly, a similar phenomenon also occurred regarding their movement orientation and exploratory behavior. Similar to the behavioral endpoint results, a comparable gradual recovery curve was also observed in the FD and entropy results. Here, the amputated fishes displayed similar FD and entropy values at 3 dpa. In order to investigate the relationship between zebrafish behavior recovery and caudal fin regeneration, the size of the caudal fin after the amputation was recorded and measured every 5 days for 30 days. From the results, the caudal fin regeneration had started to occur on the earliest observation, which was on 5 dpa, as the whitish stripe of tissue had emerged and enlarged beyond the amputation site. Then, on 10 and 15 dpa, the new tissues persisted at the distal area of outgrowth, while in the proximal part, the white tissues had progressively pigmented and re-differentiated into the mature fin fold. Later, at 20 to 25 dpa, the size and form of the fin were almost fully restored, leaving a fragile whitish material at the fin margin that may be maintained throughout the entire life of the fish. Finally, based on the literature, the regenerated fin should have regained its original length 30 days after amputation. Interestingly, based on the graph and previous behavior results, the amputated zebrafish had already been capable of performing a comparably normal behavior at 5 dpa while their caudal fin was only around 20% regenerated, indicating that the absence of a caudal fin did not majorly cause the abnormal behaviors observed on the early observation. Several previous studies had demonstrated that the behavioral changes after the fin amputation procedure are related to the pain elicited from the amputation process. Therefore, it is intriguing to observe the long-term effect and effectiveness of a commonly used drug with pain-relieving properties, lidocaine, in preventing any behavioral responses to the fin amputation during the whole recovery time. This experiment is important because, even though lidocaine has previously been demonstrated in reducing the behavioral and physiological symptoms in fin amputated zebrafish, no study has evaluated its long-term effect on the amputated fish. As we expected, lidocaine appeared to ameliorate the behavioral effects of the fin amputation in that movement complexity was almost fully restored to those seen in control fish. Interestingly, this effect lasted during the whole observation period. After further observation, we found that even though behavior recovery was observed in the rescued fish group, some significant differences were shown in some of the behavior endpoints. These differences may affect their movement predictability, resulting in a statistical difference in their entropy values. In addition, we also provide the behavior results of the non-amputated fish treated with lidocaine. Interestingly, similar to the fin amputated, lidocaine-treated group, this group also possessed different preferred positions in the tank during the test with a similar vertical exploratory behavior to the control group. Furthermore, slight abnormalities were also found in the movement orientation of this group that also may explain the differences in their entropy level to the untreated group. To sum up, there are 2 key points from this study. First, amputation in the caudal fin resulted in the most pronounced behavior alterations, and their behavior was fully recovered before the caudal fin was fully regenerated, indicating that these behavioral changes came from pain elicited from the fin amputation. Second, while lidocaine treatment could ameliorate the behavioral effects after the amputation procedure, it did not accelerate the behavior recovery process. Instead, it caused the fish to display some slight side effects. In conclusion, the results of the present study demonstrated that, by 2 days, zebrafish behaviors have not recovered, indicating a prolonged complex change in behavior and the possibility that fin amputation is actually of moderate severity unless analgesia is provided. However, one must keep in mind that, even with treatment with analgesia, specifically lidocaine, side effects could still occur in the treated fish, especially in their behaviors. Since fin clip or amputation, routine procedures to harvest tissue for genomic screening by genotyping mutant and transgenic lines established for experimentation are seen as mild under the Home Office licensing legislation, the current results highlight the importance of refinement to current protocols. Nevertheless, since we evaluated swimming behaviors only, it is intriguing to evaluate the potential effect of fin amputation on other behaviors in the future. Observing complex behavior in zebrafish with amputated fins opens new avenues for pain research. In addition, future studies assessing the behavioral effects on zebrafish after a smaller tissue removal than the current study would also be very valuable to provide a new perspective that improves our understanding of the risks of fin clips. That is for my brief explanation about one of my research. If you'd like to know more about this study, please refer to the link displayed on the first slide of the video. Thank you for your time and have a nice day.